component of what we are calling CBL's virtual science semester. Normally about this time of year, I would be standing in front of a room of almost 100 people welcoming you all to a public seminar. It feels a little bit odd standing in front of an empty room welcoming you to a public science seminar. It feels even odder wearing a jacket and tie for the first time in over 6 months. I hope you are all able to wear much more comfortable clothing than that. I also hope that I will not get the scrutiny that 2 other gentlemen who are talking a little bit later on tonight will get, although I'm happy to try and verify any of the facts that I explained tonight. So as I said, this is part of a tradition at CBL of engaging with the community. We do so through series of public seminars. We do through programs with the schools, and we do so through an open house early in September in every year. Last year, for example, we welcomed over 900 people to our campus. It is a real disappointment to us that we are unable to welcome people to our campus this year. And as we thought about what we would do in response, we thought about trying to do a one-day virtual open house and quickly decided against that. And instead, we've put together what we're calling a virtual science semester. And this is the first part of that virtual science semester. And so on behalf of everyone at CBL who has worked very, very hard in putting this together, I want to welcome all of you to CBL's virtual science semester. There will be opportunity to ask questions through the chat box at the bottom. One of my colleagues is monitoring and will moderate questions at the end of the session. And if any of you have further questions, I'll have my contact information at the end of the presentation. So let me start by sharing my screen. And as I tell all of our students, we should all be ready to answer these 3 questions. And so I'll hold myself to the same standard. I hope to start by giving you a very brief introduction about what the virtual science semester is, who we are, why we're doing it. And then for the bulk of the evening, I'll spend my time talking as advertised about oyster aquaculture and its role in the Chesapeake Bay. So let's start with that what question. Our virtual science semester is exactly what it suggests. It's a semester-long program that will be conducted virtually that focuses on science. There are 6 elements to the program. There are some behind-the-scenes videos that are supposed to replace our virtual open house. We normally can't welcome you to all areas of the campus for safety concerns. And so we've taken the opportunity to do short video tours of those areas of the campus that we don't normally allow the public into. So in the behind the scenes area, you'll see a number of short videos appearing over the next few weeks, introducing you to some of the work that we do. We are immensely proud of our graduate students, and so one of the elements is interviews with our current students, focusing on why they came to CBL, why they're interested in a career in environmental sciences, and what they hope to achieve during their graduate program. We're as proud of the alumni who've graduated from here and moved on to impactful and important careers. And so a second section are interviews with some of our alumni, reflecting on what they learned at CBL, what valuable lessons they took away from us, what valuable lessons they use in their careers. And it also showcases the diversity of careers that our students go on to pursue. The 4th element is science at home. These are a number of virtual science activities that are aimed primarily at middle school age children, some of which are curricula that were already developed, some of which are new, small sort of experiments that people can do. And we're immensely excited by that activity. The fifth is our Science for Citizens program, of which this talk is the first one, and more about that in a minute. And the final element, of course, is that we hope you support this program. You can show that support in many ways. You can show that support by learning more about the environment and what you can do to protect and sustain the environment. You can show that support by engaging with your federal, state, and local legislators and executives. And of course, ultimately, you can show that support by donating to support our graduate students. All of this information is available at this one website. If you go to the central UMCES website and just search for Science Semester, it'll bring you to this page if you don't have the link directly. So as I said, this Science for Citizens is part of an almost decade-long tradition here at CBL. For the last 10 years, we have presented 5 public seminars in the spring and 5 public seminars in the autumn. The image you see on the screen is one from last autumn where we invited ex-students back to let the community know what they are doing in their careers currently. The young lady in the middle is an ex-student of mine, Veronica Caceres, who now works on the conservation of sea turtles. She told me her mum would never believe that she came back to give a presentation, so we staged a photo that I sent to her mum just to prove that Veronica was indeed the star of the show. These presentations have covered a range of topics. We've given presentations on the ecology of the Chesapeake Bay, on water quality, on ballast water treatment. We've given talks on fisheries, whether that be blue crab or striped bass or menhaden. We've given talks that have been particularly well received on sea turtles. And whenever we talk about dolphins, we typically have to do 2 presentations because the room is so crowded. And we've also talked about environmental science more generally. We've talked about climate change. We've talked about intensive agriculture and its effect on the environment. We've talked about drug discovery from the environment. And 55 of those past presentations are available on our website. The address is on the screen. But again, if you just go to umces.edu and search for Science for Citizens, you can navigate to those presentations. They were not designed to be played over a screen. They were designed to allow our regular attendees to catch a seminar that they missed. So some of the video content is not of the highest quality, but I can assure you the science content is of the utmost quality. These presentations are made possible by the generous support of 3 local sponsors, PNC Bank, Toyota of Southern Maryland, and Team Hyundai. Normally they pay for the tea and coffee that attendees at our Science for Citizens enjoy. Today they're paying for our ability to advertise, get the word out to a broader audience. And so we thank them for their support. So who are we? We are, first of all, the kind of environmental organization that is committed to communicating our science to the community. So this is a new sign that has recently been installed in Solomons that shows the history of the water temperature at the end of our pier over the last 80 years. Every one of those stripes is the average water temperature in a different year. Blue colors, which are early on in the history, in the late 1930s, early 1940s, change slowly to redder and warmer colors until this last year, 2019, was the warmest year on our record. We do that as part of an organization called the University of Maryland Center for Environmental Science. We are the founding laboratory of UMCES. And UMCES is Maryland's graduate university for the environment. We train graduate students in environmental science to go on and have impactful careers in government, in academia, in the not-for-profit world, and in the corporate world. We have a unique legislative mission that says we will conduct a predictive ecology. And we don't honestly understand what that means, but we interpret it to mean that we conduct the science we need for the environment we want. We conduct that science in a framework that we call environmental intelligence. Just the same way as a business may undertake business intelligence to develop a business plan, or the US Department of Defense conducts national intelligence to ensure the protection of the nation, we conduct environmental intelligence by gathering, analyzing, and interpreting data to better understand and sustain our world. And the climate stripes that you see on that sign is one example of the kind of communication that we undertake. We make an impact in Maryland, we make an impact nationally, and we make an impact around the world. We build on a now 95-year tradition of excellence in research. The distinguished looking gentleman on the left of the screen is Dr. Reginald Truett, who was the founding director of CBL. He was trained as a fighter pilot in World War I. He was a champion ballroom dancer. He was athletic. He took College Park to their first national championship. He was a world-renowned expert on the biology of crabs and oysters. And I'm sad to say the only thing I have in common with him is that I'm also director at CBL. In this last year, our faculty, our 25 faculty, our 28 research staff, and 34 students have all been working hard despite these unusual circumstances. Over the last 12 months, we've submitted more than 75 proposals for new work. We've managed and sustained 49 active research research awards that bring in more than $10 million in grants and contracts. We published 81 peer-reviewed scientific papers, and we conduct state-of-the-art research on a number of topics involving the Chesapeake Bay, involving local issues, and just as importantly, involving national issues, whether that be fisheries management across the Atlantic, or whether that be working to solve the wicked challenge of why science and technology is not more diverse and inclusive. In addition to that science, we also have a tradition of excellence in service. Our faculty make an impact. We have faculty on 4 National Academies committees. We have faculty on the Committee for the UN Decade of the Ocean. We have a faculty member, Mario Tamburi, on the top left of the quadrant, who serves on the International Maritime Organization's Marine Debris Committee. This is a committee formed by the organization that governs shipping around the world. And they reach out to one individual at a small institution in Maryland. And that expertise and that service exemplifies the kind of work that we do. We have another committee member, Hallie Kilbourne, who is next to Mario on the top row, who serves on the US National Committee on Climate and Ocean Variability. Below Mario is Lisa Wanger, who's recognized in California in their California Bay Delta program, which is similar to our Chesapeake Bay program, and she serves on their Independent Science Committee. We have many people, including Mike Wilberg, who is on the bottom row next to Lisa, who serve on fishery management committees at the federal, regional, and state level. We serve on the Chesapeake Bay Trust, on the Patuxent River Commission, and we're also engaged locally in the Calvert County Environmental Commission. So we really are committed to service from outside our back door to around the globe. And of all of that, we are most proud of our commitment to education. We teach an innovative curriculum that promotes interdisciplinary understanding in our students, given a strong foundation in their core area, and they are engaged in research, solving real-world problems from the very first day they walk in through our doors, and that continues to the day they finish. And they are a remarkable group of young people. We would love for you to be able to meet them, and perhaps when this pandemic is over, you'll get that opportunity. But until then, I know that they would appreciate your support. Why is this all so important? This is a picture I always show at this slide. This is my family taken at Christmas, just outside Buckingham Palace. And it's important for the next generation. I think all of us at UMCES are driven by a passion for science, but equally for a passion to leave the world in a better place than we found it. And it is really for the next generation, for those that are coming up, for those who show such passion for the environment and such passion to sustain the environment, that we are committed to the work that we do. So with that as a brief introduction, the rest of the presentation tonight will focus on oysters. And anyone who's been to CBL over the last few years will know our seawater labs, and the area in front of CBL is full of tanks containing oysters. So the question is, what's with all the oysters? And it stems back to our founder, Reginald Truett. And I want to read a short paragraph from our collection. This is our Chesapeake Biological Publications from 1927 to 1938. This is the first contribution written by Reginald Truett, and he writes, "For fully 3 decades now, there has been an almost steady decline in Maryland's oyster production, until those charged with the industry's welfare, indeed those who have the state's interests at heart, are alarmed for its future." Now, many people are surprised that that was the condition in 1927, a condition that perhaps we find ourselves in today. And so part of CBL's commitment, part of UMCES's commitment, is to try and help understand how we can restore and sustain oyster populations for their ecosystem service, for their fisheries, and for their use in food. That quote from Truett was from 1927. It's not that long ago that I could have looked out of my window at my office at the lab, and this is a view from that window, and I would have seen literally dozens of oyster boats. This chart shows the history of landings in Maryland over the last 100 or more years, and 100+ years ago, we were landing remarkable numbers of oysters. It was a period from 1880 to 1920 that was characterized by systemic overfishing. We simply lacked the understanding of the biology of the oyster to know that there were limits to its harvest. From 1920 to 1950, there was a period of somewhat stability and a period of what I'll class management indifference, because there were no dramatic changes. From 1950 onward, we saw the introduction of diseases into the system, which caused a rapid decline to a very, very low level of oyster harvest that are now continued to this day. And the question is, what can we do to help restore, sustain those oyster populations, both for their ecosystem services and for the cultural identity that they bring to the region? So let's start with a background on the life history of oysters. We're going to start at sort of the 8 o'clock position on that diagram, where oysters spawn. They release sperm and eggs into the water. The eggs are fertilized into the water, and they transform into a swimming larva that has a ciliated fringe that enables it to generally control its position in the water column vertically, but it's so small it has no control over where tides and currents may take it. After about 2 or 3 weeks, it begins to transform into a stage that begins searching for the bottom. It begins searching for hard substrate on which to settle. And when it finds suitable substrate, sometimes other oyster shells, sometimes other hard substrates, it transforms into what is known as a spat, and then it will never move again for the rest of its life. Up until the early 1920s, we knew an awful lot about spat. We knew an awful lot about the adults, but these intermediate stages, though described anatomically and though described microscopically, we had very little understanding of how the biology played out to take adult oysters through those larval stages to produce spat. And it wasn't until 1922 when a gentleman called Wells was credited as being the first person to close the life cycle of eastern oyster. And by closing the life cycle, we mean that he developed the culture techniques to take the eggs and the sperm from oysters, to fertilize them, to culture the pelagic larvae that are swimming up in the water column, and sustain them for long enough until they settled onto shell and began to grow. And he realized quite early on, in fact, the cartoon that you see on the right is taken directly from his 1922 paper, that effectively a thimbleful of oyster larvae have the potential to become a boat full of oysters in a relatively short period of time. And that technology has matured over the 80 years since then, and is used routinely for oyster restoration. Our sister laboratory, Horn Point, on the eastern shore, produces billions of larvae a year for oyster restoration in places like Harris Creek. It has also been used for fishery replenishment. Local watermen's committees, oyster committees, take spat on shell and place them on fishery grounds. Much of that spat on shell is produced by our colleagues at Horn Point. And Wells also realized early on that there is the potential to grow oysters as a crop. The challenge in growing oysters as a crop is to keep up with exponential growth, because within a year, you go from a thimble to a boatful. And that is one of the central challenges of oyster aquaculture. Why is aquaculture of oysters attractive? There are several reasons, but perhaps first, it requires no fresh water. As our climate changes, fresh water becomes scarcer, and almost all of our intensive agricultural methods require fresh water, and that will become increasingly scarce. Also unique for aquaculture is that because oysters eat single-celled plants in the water column, eat phytoplankton, you don't have to add additional food to the tanks. With fish and many other invertebrates, you do have to find a source of food for those animals, and very often it has to be animal food because plants don't contain the essential amino acids that many animals need to complete their development. I will note that our colleagues at our sister lab in Baltimore at the Institute of Marine and Environmental Technology have developed methods to raise plankton that contain those essential amino acids, thereby providing the potential for a plant-based food for aquaculture, and that's a significant breakthrough. But as I said, because we go from a thimble full to a boat full, it requires large amounts of leased bottom to allow the oysters to grow. And that can produce conflicts with other users who are interested in using that area of the Chesapeake Bay. And we'll explore that a little bit more later on in the presentation. So there are three main forms of aquaculture as practiced in the Chesapeake Bay. The first and the oldest is what is known as direct placement on the bottom. And in this situation, the grower would take spat on shell from a hatchery. They would put them directly on the bottom, preferably on preferred bottom, such as relict oyster reef or existing oyster reef. And they would allow those shells to grow and to enter the fishery and be harvested in a normal way. This technique is used, as I said, by county watermen's committees. It's used also as a managed reserve in the Potomac River, and it does require some kind of ownership of the bottom, or at least some kind of reduced access to the bottom. The next two types are much more by way of agriculture, in the sense that they use structures to constrain and hold the oysters so that they can be moved and handled easily. And they differ primarily by where in the water column the oysters are placed. In bottom cage gear, as its name suggests, the oysters are placed in cages or bags, and they sit on the bottom or just off the bottom. This also requires leased bottom, but as you can imagine, once you place them in cages, it provides a lot more control of access to those oysters, either in prevention of poaching or when you want to harvest them later on. And the other main type of aquaculture is floating cage gear. One of the challenges of bottom cage gear is that water quality at the bottom can sometimes be poor. You may have heard of low oxygen conditions in the bottom waters of the Chesapeake Bay. And so some growers place their oysters in floating cages that bring them well up off the bottom so that they are suspended in the water close to the surface of the water. The other big breakthrough for aquaculture, which I've not mentioned yet, is that often it involves triploid oysters. Triploid oysters are sterile. They have an extra set of chromosomes that prevent them investing energy into reproductive organs. So all of that energy is rather invested into growth. And because they're not investing in reproductive tissue, they can be harvested at any month of the year. And that opens up the potential of harvesting oysters in months that don't have an R in their name. So just to give you an idea of what oyster aquaculture can look like, this is a short video looking out on an oyster farm in St. Mary's County. And as the film plays, I'm going to pan to the left. And what you're looking at is at low tide, you're looking at some of those bottom cage gear just poking their head up above the water. So you can see some of these oyster farms can be sizable. And there are probably 1,000 or more cages on that oyster farm that each contain in themselves hundreds to thousands of oysters. So just to give you a picture of what this looks like, The bottom cage gear is the single picture on the left. That's one cage that probably holds 6 to 9 bags of oysters. To give you an idea, the cage is the size of perhaps a medium-sized end table or a footstool. On the upper right side are examples of both. You can see you can see the bottom cage gear here at the bottom of the screen and the floating cage gear at the back. And then at the bottom is an example of a more sophisticated floating cage that I'll talk about again very briefly at the end. So what does oyster aquaculture involve? It involves everything that you would think of as normal farming. It involves the management of the brood stock. It involves producing the oysters and setting those larvae onto some kind of substrate. Those first 3 processes often occur in a hatchery. Once those larvae are set, they need to be cared for, or called husbandry of seed. And those 2 functions, setting of seed and husbandry, sometimes occur in operators that run nurseries. And then once those young oysters have started growing, they are cared for and grown out, harvested, delivered to market in commercial operations. Now, the challenge here is that there is considerable investment in expertise, time, and finances in running the hatchery. But there is little value in the product that the hatchery produces. This means that many hatcheries become vertically integrated in that the hatchery supplies its own nursery and its own nursery supplies its own commercial production, such that those larvae are often not available out to people who just run nurseries or just run commercial production operations. So larval production is one of the principal challenges to both oyster restoration and aquaculture. And the challenge is made worse by this transition. This settling of larvae has generally a low success and a highly variable success. So one of the first challenges we were asked to address is, well, what should an operator of a nursery or a commercial production farm do? Should it just buy any larvae it can obtain, or should it be more selective? This work was funded by a unique program in Maryland called the Maryland Industrial Partnership Scheme. In this scheme, the state provides money to academia and any small business that wishes or that has a problem that it thinks universities can solve. They develop a collaborative proposal, and if it is successful, the small business funds 10%, the state funds 90% of the first 100,000, and it can grow to a phase 2 beyond that. So we were approached by a small oyster farm in St. Mary's County who wanted to understand whether or not they should buy larvae whenever they could get them, or whether they should be more selective. So the experiments I'm going to describe briefly occurred at CBL in 2018, were conducted with my collaborator, Dr. Karis Mitchellmore, and with the considerable help of my technician, Skylar Galt. We conduct firstly nursery trials. These are conducted in silos that produce a downwelling flow. Each one of these silos is formed by 2 5-gallon buckets that are cut in half and then glued together with Nitex screen between them. And the flow comes from the top gently into the bucket, through the mesh and out the bottom. And by controlling the height of water in this larger outer tank, we can control the flow rate in the inner tank. And we followed larvae for between 8 to 13 days, and we estimated settlement. We also tried larvae from multiple different hatcheries, and we tried larvae that had been intentionally stressed, as well as some that had been grown in optimal conditions. This is what the larvae look like as they settle. You can see in some of them the oyster larvae shown here, and the small grain of oyster shell known as microculture on which the larvae have set. And each larva generally sets on one grain of ground up oyster shell. So these are the results. Overall, we experienced about 35% settlement success, which is somewhat typical for many nurseries. But the important thing is between batches, we had experiences of between 3% to 100% settlement success. And so the chart shows different hatcheries in different colors. So red is hatchery 1, batch 1. Green is hatchery 2, batch 2. Cyan is 3, and batch 4 is in purple. And then the two different bars are one in which the larvae were set in what we think of as an optimal condition. They were provided ambient water. And in the second one, they were starved. We filtered the water through a 5-micron filter, effectively removing all of the food from it. And you can clearly see both hatchery and the condition of the larvae matter on their settlement success. So what happens then when we have now created larvae that are different conditions, of different health? What happens when we grow them out? These trials were conducted also in 2018, but went on to 2019 in larger tanks that are about the size of a 55-gallon drum, but they have mesh on the bottom, and water flows into the corner of this big tank through the bottom of the silos and out through the side. And so by controlling the flow of water in these tanks, we can control the flow of water through the mesh at the bottom up past the larvae. And so in many ways, these oysters are bathed in a very rich soup of food, and we would expect them to grow and survive well. And so we monitored growth and survival for 8 weeks in these systems. These are what the young oysters look like after about 2 weeks. These are probably about 1 inch in size, so about 20 millimeters in size. These are all triploid oysters, and this is just a selection from one tank. You can already recognize them as oysters. These are their growth, again, showing the 4 different batches. The first line is the starved, or the treatment where they were starved as larvae. The second one is the treatment where they were given the best possible conditions as larvae, and then the 4 different batches. If you remember, batch 2 had remarkably high settlement success, but perhaps you can already see that they may have set with a high success. They didn't necessarily grow as large in the same time frame as larvae from batches 1, 2, and 3. Overall, our oysters grew almost 20-fold in the 8 weeks, and they grew at different rates. But the challenging question is which one grew fastest? And in fact, it was those oysters that had experienced the poor conditions during their settlement phase that grew significantly faster than oysters that had enjoyed optimal conditions throughout their husbandry. And in a sense, oysters are compensating for early poor conditions by growing faster later in life. So that if we look at the overall yield from those 4 different batches, although settlement success and size at settlement varied almost 40-fold, the final yield to the grower only varied threefold. So biological processes of growth and survival dampen the inter-batch variability such that it probably isn't worth the grower being too selective about where they take their larvae from. Rather, they should take larvae whenever they have access to them, and importantly, space to grow them. After those grow-out tanks, all of the oysters went out to leases in front of the lab in bottom cage gear. Underneath each one of these floats, they're not seagulls in the water, they are oyster floats. Underneath each one of these floats is a cage containing 9 bags of oysters, and we tracked those growth of those oysters for another year or more, and I'll talk more about that at some later time. So what about trends and challenges then? So aquaculture for oysters is a small but growing component of the Maryland oyster industry. It has accounted for between 4 to 20% of the total harvest of oysters over the last 5 years, and that fraction has grown steadily. Since 2018, there have been 105 applications for leases for aquaculture. So clearly there is an interest in developing more aquaculture within the state, and there appears to be considerable potential for growth for those for access to capital, larvae, and leases. So who benefits? Clearly the oyster grower benefits. This is an industry that has the potential to provide sizable profits. Diners benefit from Solomons to Seattle. One of the remarkable features of oyster aquaculture is that oysters can be harvested from the Chesapeake Bay early morning in St. Mary's County, be on a truck to Baltimore, and before noon be on a plane to Seattle and on a diner's table that evening in Seattle. It truly is a national scale industry. Does the Chesapeake Bay benefit? Well, it certainly benefits from the filtering capacity that the oysters grown in aquaculture exert, no differently than the oyster reef. Do oysters in aquaculture reduce nitrogen levels in the Chesapeake Bay? Possibly, more likely, probably, but there's still some uncertainty as to the extent that that occurs. Do they create habitat? They certainly do. That oyster farm that I showed you the video of is certainly a habitat for crabs and other shellfish and striped bass and croaker and other animals. But that habitat is obviously not permanent. It is withdrawn at regular intervals. So if there are people who benefit, who loses? Clearly, perhaps those who are preempted from the space. That may be watermen. It may be waterfront property owners who don't like the thought of an oyster farm in front of their view. It may be recreational boaters who worry about becoming entangled in lines. Does the Chesapeake Bay lose? Potentially loses because the habitat is temporary rather than a permanent, fully established oyster reef. And oyster farms certainly don't provide the coastal resilience that a permanent, fixed, fully functional oyster reef presents. So there certainly are benefits, and there are certainly some losers as well, or losses. How do we manage them? We undertake what's known as marine spatial planning. This is a map from the DNR aquaculture siting tool showing the area immediately around Solomons. For those of you who don't know the area, Solomons is right where my pointer is now. In fact, I'm speaking to you from a room at CBL right where the pointer is. And in the water, you'll see a myriad of different colored zones. The dark red stippled area are what are known as public shellfish areas. They are the original Yates bars. The purple areas are areas of restoration. The bright green area there you see opposite Patuxent Naval Air Station is an oyster sanctuary. The light gray lines are places where pound nets are licensed. And the light blue, and the light green, just in front of the lab here and over on Drum Point are areas of submerged aquatic vegetation. And the light blue are areas of oyster leases for aquaculture. And there are 4 of them in this picture. There are 2 just here by us at CBL. There's one on the opposite shore of the Patuxent River that is a little bit larger, and one quite small further down. As you can see, there's not a lot of open space. A lot of the nearshore space, which is where oyster aquaculture and oyster bars like to be, is already allocated. So how do we go about allocating that limited space? Well, happily, for me, that's not a science decision, that's a socio-political decision. Science can inform the outcomes of alternative options, but it is not the solution to the problem. Perhaps one of the things as market decisions, what does the market value? And perhaps one of the solutions is a technological solution of potentially moving oyster aquaculture into deeper waters where space is less constraining. And that more complex floating aquaculture cage that I showed you was one of those efforts to try and explore a technological solution. And it's another MIPS funded project that we've worked with an engineering firm. So that's it for the presentation tonight. If you want to know more about my research, you can find any of that I've talked about on that page. You can also contact me from that page. I'll encourage you to join us for the next 4 weeks for other Science for Citizens presentations. Next week, my colleague Dave Secor is talking about striped bass. The following week, Ryan Woodland is going to be talking about our work on the Patuxent River. The week after that, Jeremy Tester is going to be taking a more historical view to the Patuxent. And then finally, Dr. Helen Bailey is going to be talking about plastic waste and pollution in the Chesapeake Bay. And so with that, I will finish, and I'll be happy to address any questions that have occurred in the chat box. And so, Sarah, I'll turn it over to you, if I could, please, to try and moderate the questions.
Good evening, everyone. I'm Sarah Brzezinski. I'm the Outreach Coordinator at the Chesapeake Biological Laboratory. As Dr. Miller mentioned throughout the seminar, I've been keeping track of the questions that have been posed. I'd invite any additional questions to be submitted through the chat box at this time. To kick things off, Dr. Miller, here's our first question. You spoke about the filtering capacity and denitrification potential of oyster aquaculture in the Chesapeake Bay. Can you explain a bit more about how oyster aquaculture appears to impact water quality in the Chesapeake Bay?
So this is an area of active research because it is clearly, as aquaculture is growing, it becomes an important question. Oysters grow by filtering suspended material out of the water column. And so the very fact that they are filtering material out of the water column is a positive benefit. The fact that they lock some of that organic material and some of that nitrogen up in their tissues, and then we then take that material out of the water column, is also a benefit. But one of the things we've learned in the last decade or so is that a lot of the nitrogen removal that occurs on reefs does not occur as a direct result of the oysters filtering, but occurs in the microbial communities that are in the cracks and crevices in the oyster reef itself. And it is not fully clear yet the extent to which oyster aquaculture fully captures and fully represents the potential for nitrogen or denitrification that a mature oyster reef presents. So they clean the water, they lock some of the suspended material up in their tissues, which we then remove. But whether they are as effective as a mature oyster reef is still an area of active research.
All right, our next question is, are there any natural oyster reefs left in the Chesapeake Bay, and if so, where?
There are lots of natural oyster reefs left in the Chesapeake Bay. I certainly, if I gave the impression that somehow they have completely disappeared, I was wrong to give you that impression. There are still a large number of oyster reefs throughout the Chesapeake Bay. They are simply not the extent that they were in the 1800s and earlier, when in fact they were so extensive and so large that they almost protruded out of the water at low tide and became navigation hazards for shipping. That period of intense overfishing that occurred from really 1880 to the 1920s removed a lot of that oyster biomass, but they are by no means extinct. There are still fully functional oyster reefs in the bay, and there is a lot of work to restore them.
Do we export oysters internationally?
I honestly don't know the answer to that question. My guess is probably perhaps to Canada, but probably no further. Oysters have to be eaten fresh, or at least on the half shell they have to be eaten fresh. When they are shucked and canned, perhaps so, but I would imagine that's a fairly small export, but I suspect there's someone on the Zoom meeting who will correct me at some point. So only a small fraction, I suspect, goes internationally.
Our next question is, when and how will Maryland catch up with Virginia's number of nurseries?
That's a great question. The aquaculture in Maryland was really given an impetus about a decade ago. State government worked to try and promote oyster aquaculture and promote the formation of oyster farms. But as I said, the value in oyster aquaculture is in the oysters themselves. And so people have been less eager to set up new hatcheries. There is some emerging technology that I think will make that more possible. And there are some large investments on the Eastern Shore to try and set up another substantial hatchery. So it is recognized that there is a shortfall of hatcheries, and I think it is just a case of developing the business model that makes them profitable.
All right, and another question. Do younger oysters filter water as well as mature oysters? In other words, could it be more beneficial for the bay to have old beds of oysters over young beds of oysters that are being harvested constantly?
That's a great question. Oysters, the amount an oyster filters depends upon its size. And so there is a trade-off between filtering rate and size. So if you allow oysters to get large, they filter more oysters. But there are probably fewer of them that survive to be large just through natural mortality. So the peak of filtration is probably in the middle size classes of oysters, even on a natural oyster reef. What's different between aquaculture and the oyster reef is all of the other filters that are associated with the mature oyster reef that are less common in the aquaculture cages, in the aquaculture system. So overall, the filtering success of aquaculture is probably less than a mature oyster reef itself, but not really because of the size of the oysters.
What about using creeks? Town Creek, for example, has ample waterfront and deep water that were colored pink in the chart you presented.
Yeah, some of those areas, and I should have highlighted them, some of them are colored pink because those areas are closed to commercial oyster harvest, whether it's through wild fishery or through aquaculture, because of a relatively higher risk of disease in the oysters that can harm humans who eat them. So oysters, sorry, not disease, pathogenic animals that enter the oysters that cause harm to humans. Often harmful algal species that cause extreme digestive stress, and in their ultimate form can be fatal in some rare cases. And those diseases, those harmful algal blooms, are more common in the warm, shallow water that is not flushed as regularly by the tide as they are in the open water. So you could certainly put oysters in those creeks to try and help filter the water, but there is less of a likelihood that those oysters could then be used on the commercial market.
Does oyster aquaculture compete against watermen, not just in terms of bottom use, but actual supply and impact on market prices?
So the economics of that is beyond my knowledge, to be fair. They are slightly different products, right? The aquaculture because as I showed in the picture, the oysters set on individual grains of shell. When they grow up as individual single oysters that can look really pretty on a dining room table. So if you go to the Ritz-Carlton and they want to serve you oysters, they are going to put a premium price on these individual oysters that come from aquaculture. If you are shucking, you may prefer wild oysters that have often larger sizes. And so they're really competing in different parts of the market. And I think each sector begins to specialize in one area or another. And I suspect just as we see in people who appreciate fine wines or people who appreciate cheeses, those who really appreciate eating oysters begin to develop preferences or tastes for perhaps a wild oyster or perhaps an aquaculture oyster from a certain place. For those of you who've eaten them on the half shell, it is very easy to tell a Chesapeake oyster from a Long Island Sound oyster simply because of the saltiness of the oyster. So they are, I think, dividing the market somewhat differently.
Your summary suggested that further development of the oyster aquaculture industry in Maryland is related to shoreline planning. Outside of that conclusion, what key scientific constraints are there to growing the industry?
So I do think shoreline planning is a critical part of it. There are limited areas in which oyster aquaculture can occur. It has to fit around existing uses, and it typically occurs in shallow water, which keeps it into the shoreline. And so there has to be accommodations among the different sectors that wish to use that portion of the shoreline, whether that's a marina that want access for their boats to be coming in and out, and they're worried about oyster aquaculture operations near to the channels that their clients use to access the marina, whether that's existing wild oyster bars that are exploited by commercial watermen, or whether it is a homeowner who wishes to have a bucolic view out from their veranda and not one with floats. So there are conflicts. There are conflicts that I think can be easily resolved, but they are, as I said, it's not a lacking in science question. It is a socio-political question of what we as a society wish to promote.
And are there any strategies that you think we should use to overcome those obstacles?
Having just said it's not a science question, I will avoid treading on the very thin ice and the limits of my knowledge to try and suggest solutions. I do think there are some potential technological solutions that would access deeper water. That's one area that might work. But those technological solutions have costs, and whether they become economic to do in deeper water is open to question. But it is a case of people who wish to use the same areas for different uses that are in conflict have to be able to work together to come to a resolution. And that's not a scientific question.
From whom is space leased for oyster aquaculture in Maryland?
So effectively, it's leased from us, the citizens. So DNR is charged through the state constitution with the ability to lease public space for private use. And it is effectively public space that society has chosen to temporarily put into a private use. This is not permanent. It is not that the grower owns the land. It is that they have leased that portion of land for a period of time that they are going to use it to produce oysters. When they stop producing oysters on that lease, the lease closes. So it is not in the sense that a farmer owns a farm field, and if they choose to stop farming, they still own the farm field. This is the case where a grower leases a piece of bottom, and if they stop using it to produce oysters, that lease terminates. But effectively, they are leasing it from us, the citizens of Maryland, and that is why I said it is a socio-political problem rather than a science problem.
Can you describe the solar-powered float platforms and how they were used?
So this is a technology that's being developed by a private company. They own the intellectual property for that, and it's probably not fair for me to divulge that technology, but it was a very creative way to try and expand where aquaculture can occur.
What's the seed capacity of wild oysters in this area? Setting aside the question of oysters being seeded and farmed, has it recovered in any sense?
So oysters still reproduce naturally in the environment. There are occasions where there are substantial sets of spat, sometimes over broad areas, sometimes over more restricted areas. And there are some parts of the system that seem to be more reliable than others. So for example, the St. Mary's River seems to be a reliable producer of oyster seed. And one of the interests, certainly among the commercial watermen, is to try and find those areas that are reliable producers of seed so that they can be used, so that the seed produced from those areas can be moved to try and kickstart oyster bars elsewhere in the bay. So there is natural reproduction, but if you remember back to the chart that I showed early on in the presentation of the oyster harvest, you can imagine that harvest roughly parallels abundance. And we are at a fraction of 1% probably of the historical abundance of oysters. So you can only imagine the reproductive capacity has declined sharply.
Are there certain areas of the Chesapeake Bay that are better for oyster farming? You spoke briefly about some areas being better for seeding. What characteristics go along with oyster-friendly areas?
So I think like some farmers have a particular attraction to certain types of farm fields, the oyster growers I've talked to have attractions to certain types of bottom and certain types of system in terms of how readily they are flushed or how far they are away from development and other potential sources of contamination. So growers are very particular. They look for often high flushing rates so that there is a low risk of their oysters picking up these harmful algal blooms. They look for hard bottoms. They don't have to be shell bottoms or rocky bottoms, but they have to be hard enough that the cages won't sink in. And they have to be an area where there is sufficient phytoplankton in the water column to feed the oysters. There is also a general gradient of disease. I mentioned that oyster diseases came in the 1950s, and diseases tend to be more prevalent in higher salinity waters. So there is a search for trying to find a sweet spot, because oysters also do better in higher salinity waters. There is a tension there between finding a place where the salinity is sufficient to support the growth, but not so high that the risk of disease comes in to kill the oyster.
Are there any collaborative undertakings between the state of Maryland, oystermen who are doing commercial harvest, and those practicing aquaculture? And what does the state do to facilitate this fishery?
So I can only talk, or I can talk directly about our efforts. The program that I've talked about, funded by the Maryland Industrial Partnership, is with one grower in St. Mary's County. They're using land in front of the CBL, and the grower and commercial watermen from Solomons are collaborating on exploring aquaculture as an alternative harvest approach for commercial watermen as well. So there is lots of discussion. There has been in the past some suspicion, but I think those suspicions are beginning to decline somewhat. And I think people of goodwill can certainly find solutions so that both kinds of industry, harvesting wild oysters and aquaculture, can occur together.
Will global warming of the Chesapeake and other waters have an impact on nurseries and natural oyster beds?
What a great question. So it is abundantly clear that our water is warming. The climate stripes that I showed during the presentation and before the presentation started show that we are some 3 or 4 degrees C warmer now than we were 80 years ago. That's a substantial increase. Although you may only just be able to tell the difference between two basins of water that differed by that temperature if you put your elbows in them, the animals can certainly tell the difference. These are what used to be called cold-blooded animals, what we call poikilothermic animals, and then metabolism varies directly with the temperature. So as the water warms, oysters can feed more rapidly, they can grow more rapidly, they can mature more rapidly. So one would expect that as the climate warms, oyster production rates will increase. And certainly if you compare oyster production rates in the Chesapeake to some of the well-known beds along the Gulf of Mexico, those production rates are higher in the Gulf than they are in the Chesapeake. So at one level, climate change is a good thing for oyster growth, provided that the phytoplankton that they rely on are also there in sufficient abundance.
Do clams and oysters live together friendly?
I think I see no reason why they shouldn't. They don't often, clams often live in softer, sandier soils, and so they don't coincide. But there is no reason to think that they wouldn't be able to coexist on the fringes of oyster reefs, for example.
All right, so we have 4 more questions. Keeping an eye on the time, I'm going to go ahead and close the chat feature to additional questions. Our next question is there a significant difference between male and female oysters in terms of growth?
Another great question. There's certainly a difference between the triploid oysters that those experiments were conducted with and diploid oysters, that is, natural oysters that have 2 sets of chromosomes, just like you or I do, because they are investing energy in growth. One of the expressions in biology is that sperm is cheap. The male gametes are small and do not require as much energy to produce as the female gametes, the eggs. And so in general, females invest more reproductively than males do.
Is there a way to leverage the aquaculture production principles to develop micro refuges, things that property owners along shorelines could use, advancing these techniques to provide ecological services?
Certainly there is. Certainly someone asked the question of Town Creek and whether that could be used. If the only goal was a water quality enhancement, that is certainly feasible and possible. If the goal is harvest, then those smaller areas face challenges because of the harmful algal blooms. But in all cases, if you are going to be placing sizable numbers of oysters on the bottom, you need permission from the state to do so. I certainly don't want to encourage people to start pouring thimblefuls of oyster larvae off of their dock.
Do we know how human digestion and human health copes with triploid oysters, which are genetically modified?
I can talk from personal experience. Mine seems to cope perfectly well with triploid oysters. They are no different functionally. They filter the water in the same way. They store energy in the same way. For those of you who visited us on an open house, my usual comment is I've never worked with an animal that poops as much as an oyster does. Triploids poop exactly the same way that diploid oysters do. These are not genetically engineered animals. They are animals that simply have an extra set of chromosomes. So I have no concerns over the potential impact of this either on the ecosystem or on people who choose to eat triploid oysters. That itself, I have no concern over.
Our final question of the evening addresses the Harris Creek project. Do you have any information that you can share about where the Harris Creek project currently stands and if there are any updated results?
So Harris Creek, in terms of the primary restoration effort, has been completed. The state still has efforts to complete some other tributaries around Harris Creek, and the state has selected 5 new tributaries for restoration. The monitoring of Harris Creek is ongoing, and I'm ashamed to say I don't have that information off the top of my head, but if whoever asked that question wants to contact me offline, I will be happy to provide them with the information.
All right, that was our last question of the evening. Dr. Miller, thank you so much for presenting an excellent seminar.
Thank you, Sarah. I want to thank all of you who have joined. I hope you enjoyed this somewhat different way of bringing science to citizens to you all. I encourage all of you to attend any of the next 4 presentations, starting next Tuesday with Striped Bass are Built for Success, and to also explore the other elements of our virtual science semester. So thank you all, and I hope you are not too tired to watch the presidential debate tonight. Thanks very much. Bye-bye.